Before a cell can divide to become 2 and 4 and 8, the DNA molecule in each dividing cell must be doubled so that the new cell receives a perfect copy. This means that, first, the double strand of DNA must be separated into single strands, and then complementary nucleotides must be linked along each of the separated strands to make two identical double-stranded DNA molecules. This doubling of DNA takes place in the nucleus of every replicating cell. The characters in this important drama are the original DNA double helix itself, many copies of 4 different nucleotides floating within the cell's nucleus, and 7 hard-working proteins dedicated to the work of making perfect copies of both strands of DNA. The same team of proteins is found in every self-replicating organism. The team copies DNA with an accuracy of only one mistake every 100,000 or so nucleotides. In step 1, protein 1, the initiator, finds the place to begin copying and guides protein 2, the unzipper, to the correct position on the DNA helix. Step 2, with the help of protein 3, the untwister, the unzipper separates the DNA strands by breaking the weak bonds between the nucleotides. The initiator exits. Step 3, then proteins 4, the builders, arrive to assemble a new DNA strand along each of the unzipped strands. Step 4, they build by joining individual nucleotides to their matching complements on the existing strand. Free-floating nucleotides bring in their own energy to create new bonds. As each new nucleotide is added to the growing chain, its phosphate bond energy goes into making the new bond to the preceding sugar. The upper builder follows behind the unzipper. Yet the lower builder, like the upper one, must build by bonding the phosphate end of the incoming nucleotide to the sugar end of the chain's backbone. She solves this directional problem by making a loop and building along the bottom of that loop. When she finishes a length, she lets go of the completed end, grabs a new loop, and continues linking nucleotides along a new stretch. So, while the top new strand is built continuously, the bottom new strand is assembled in short lengths. Lengths which are then spliced together by the stitcher. This reaction requires energy supplied by ATP. So, to review these nuclear events, we see the initiator arrive to direct the unzipper to his spot, the untwister working to help open the DNA into 2 single strands, the builders adding nucleotides to the opened strands, while the straighteners keep everything accessible and orderly. We are on our way to a new cell.